grade 12 learners, welcome to today's life science lesson. My name is Shabnam Suknandan. This lesson is brought to you by the Gauteng Department of Education in collaboration with Saibono Discovery Center. So today, learners, we're going to be looking at one of the second last topic for the year, and that deals with plant hormones. So let us start off here. Plant responses to the environment is actually dealing with your plant hormones, okay? So let us look at what is the actual weighting and the mark allocation of plant hormones in your paper 1. So plant hormones are found in paper 1. And you can see that plant hormones over here is in term 3. It's responding to the environment, and it takes 9% of your paper 1, which is a total of 13 marks. So it's a very short section, and if you study it very well, already you'll have 9% in your pocket, and that's a high percentage. So learners, make sure you take at least one day and you master everything relating to plant hormones. Now in the exam guideline, you find we have your plant hormones here, and there are 3 plant hormones that you need to know. There are many other plant hormones, but the 3 that you need to know for the exams is your auxins, your gibberellins, and your abscisic acid. Make sure that you know the spellings of these 3 plant hormones. Auxins, gibberellins, that means there's 2 B's and there's 2 L's, okay? It's gibberellins. So 2 B's and 2 L's, that's gibberellins. And abscisic sounds like scissors, abscisic acid. Okay, and then we're going to look at how do your hormones act as weed killers or herbicides. So hormones also act as weed killers or herbicides. Okay, so now we're also going to look at the role of auxins in geotropism and phototropism. Right, then we're going to look at what are the actual defense mechanisms that plants use to protect themselves, and that is chemicals and thorns. So the 3 hormones are auxins, gibberellins, and abscisic acid. Then how do hormones act as herbicides or weed killers? And then the role of auxins in geotropism and phototropism. That's where our experiments come in, and we're going to look at how plants defend themselves against predators using thorns and chemicals. Now, what does the exam guidelines say? It says, first of all, you need to know the functions of auxins, gibberellins, and abscisic acid, right? The role of auxins in geotropism and phototropism, and you need to know the role of thorns and chemicals as a defense mechanism. And then you need to know the experiments relating to phototropism and geotropism, where roots are positively geotropic and shoots are positively phototropic. All right, and that stems because they don't grow towards gravity, but they grow upwards. So stems are actually positively phototropic, but they are negatively geotropic. So stems are negatively geotropic because they don't grow towards gravity, they grow towards light. Right. So now here are the 3 categories of your plant hormones, and you learn them like this, that they are auxins, gibberellins, and abscisic acid. Right, what are plant hormones? First of all, learners, plant hormones differ a little bit from the hormones that we look at in animals. They say plant hormones are not actually true hormones because the part where they are produced, that is where they react. And you know, in terms of all the hormones in the human body, that is your ADH, your adrenalines, they are produced in one part, but they have the effect away from the part where they are produced. So they go to target organs. So animal hormones or human body hormones are regarded as true hormones, but they say plant hormones are not true hormones because they act in a part away from where, actually they act in the part where they are produced. So plant hormones, if they produce in the tip of the stem and tip of the root, that's where they have the effect. So So plant hormones allow plants to respond to certain stimuli. So what do plant hormones do? In stems, they will ask the stem to respond to light. And in the roots, the auxins in the root will cause the roots to grow towards gravity. So gravity and light are stimuli. One stimulus, gravity is a stimulus, light is a stimulus. Together, plural form, they are called stimuli. So plants must grow towards a stimuli. Right. So auxins or plant hormones allow the plants to grow towards a light source, and when plants grow towards a light, it's called phototropism. Remember, the word photo means light and tropism means bending towards, bending movement. So plants will grow towards light, that is phototropism. Also, plant hormones allow seeds to germinate in spring. So at a certain time, an appropriate time, the seeds will germinate when it's warmer, when there's water, when there's rainfall. And who allows the seeds to germinate are specific hormones. We're going to know the functions of each hormone just now, but now we're just giving a general overview of what plant hormones do. They allow the plants to grow towards light. They allow seeds to germinate in spring. They also cause seed dormancy in winter, where seeds are inactive or they are dormant. They do not germinate in winter. And this is very important because winter is a cold month. Winter, there's no rainfall, there's no water. So if seeds should germinate, they would die, and it becomes very, very wasteful for the farmer. So it's very important. Naturally, seeds become dormant in winter because of a hormone. Also, you find that as soon as spring comes in September, the peach trees and the orange trees will automatically blossom. All the little flower buds will begin to bloom, right? Who tells these buds to actually bloom and to sprout? It's actually the duty of the hormones. And we're going to look at exactly which hormone does exactly what function. So let's look at the function of auxins. Now here you can see in a shoot, this is a shoot, auxins will actually cause cell division. They also cause the movement towards a stimulus, right? They can cause a movement of the stem, they can cause a movement of the root, that is auxins. They can cause it to move towards light, right? You can see the shoots are growing towards light. And then also auxins, when they are present at the stem tip, they will also inhibit or suppress lateral branches. So when auxins are present, they will at the tip of the stem, they will promote upward growth and inhibit sideward growth. So you find auxins are actually going to suppress, as long as auxins are present in the tip of the stem, they will suppress or inhibit lateral branches from growing. Right, so let us just summarize the function of auxins. Auxin stimulates cell division. It causes cells to grow faster in roots and in the stems. It will stimulate cell elongation. Look at this. All these cells are not just dividing, not just cell division, but they are growing very, very long. Okay? So it causes cell elongation, and because of that, the stem will grow in length. Because of cell elongation, the root will grow in length. So auxins cause cell division and cell elongation at the tips of the stems and the tips of the roots. They also cause the development of fruit. It causes the oranges to start growing, the grapes to grow as well. So development of fruit is also due to auxins. Also, auxins, right? They play a role, well, not actually in the position of leaves and fruit, but hormones generally cause leaves to fall and fruit to fall. That is actually abscisic acid. The word comes from abscisic acid, means abscission. Right. Auxins also stimulate the adventitious root. So if you have a little leaf cutting, if you break certain leaves and you put them in water, you'll find small little roots will appear, right? And that is because of auxins. So auxins actually stimulate the development of adventitious root from leaf cuttings and stem cuttings. They cause tropic movement in stems and roots. And As long as auxins are present in the tip of the stem, they would cause the stem to grow apically, upwards, straight up, as long as they are present. Right, now we're going to look at the use of auxins in agriculture, in farming. So when you read metric, probably you want to actually go into the agricultural field, then you need to know the functions of these plant hormones. Right, the main factor about auxins is that they control weed growth. That means a high concentration of auxins. Farmers can use a lot of auxins and actually spill them onto your weeds, and they act as a weed killer. So they will kill the broad-leaved weeds, but they wouldn't kill your crop plants. Okay, so this is very good because by using these auxins, you're actually saving on labor, human labor. So instead of people coming and pulling out weeds, and it's very time consuming, it's very, very tiring, you just have to use the auxins as weed killers and spray them onto your weeds. The weeds will be killed, and then your crops are saved. And also, because you're using auxins and not an actual pesticide or a herbicide that is harmful, you find that the auxins will protect the good plants and they are not toxic to the environment. So the auxins will be non-toxic, the non-toxic herbicide. So the herbicides can only kill weeds. What is the advantage of using auxins as weed killers? They are non-toxic to animals, okay? And they're also non-toxic to the soil. They will only kill the weeds that you spray them on. Okay, and then you find that because you're going to be using auxins to kill the weeds, you're going to get a higher crop yield. Remember, when they give you a question on explain the advantages of auxins, right, in agriculture when they are used as a weed killer. So what is the advantage? If they ask you to explain, you got to give a statement and a reason. So auxins, a high concentration of auxins act as weed killers, right, and therefore it saves on labor. High concentration of auxins has an advantage as it's non-toxic, therefore it would not kill the useful plants and animals. So that is how you explain an advantage. So there's no need for weeding the garden because you're going to be using auxins as a weed killer, and therefore it saves on labor. You don't have to pay the humans now to pull out the weeds. Auxins act as herbicides. So anything that kills a weed or a plant is a herbicide. Remember, if you're using Doom or Raid, then that becomes an insecticide to kill insects. All right, if you're using, for example, Ratex, that is a pesticide. But when you use a chemical hormone to kill the herbs or the weeds, it's then called a herbicide. And we can use auxin as a herbicide, but it is also very expensive. So sometimes farmers opt to rather use people to put off the weeds because the auxins are just as expensive, and they might as well just pay the laborers. So how do auxins work? They will only kill broad-leaved weeds, leaves that are broad-leaved. So your mealy plants, which actually have your parallel vein leaves, they won't be killed. It's only all your big or your dorsiventral leaves of weeds. Those plants will be killed. So the weeds are killed, but not the maize plants. And here you can see a low-lying plain actually spraying, right, auxins over the maize, so the maize will be protected, but any weeds in between will be killed. Now we're going to look at phototropism and geotropism, and these are the experiments that normally come out as an investigation in paper 1. So let us look at this little pot, and you can see there are seedlings with little leaves growing, and there is a light, and you can see, learners, in this diagram here or this picture, the little seedlings or the shoots are growing. They're actually bending towards light. So what did we say bending is? Tropism, and they're growing to light. It's called Phototropism. So this is a positive movement towards the stimulus. Stems must grow towards light. So therefore stems are showing a positive response. They are growing towards the light stimulus. Okay, here you can see this pot plant fell onto its side, the wind dropped it, and although the pot plant fell, the stem will still bend and grow towards light. So the stems are showing again positive positive phototropism, right? So when it grows towards light, we talk about phototropism. And it is the stems that are positively phototropic because they move towards light. So even if the plant falls down, the stem will still bend and grow upwards towards light, and that is positively phototropic. But they are moving away from gravity. So gravity is also a stimulus, but the stem is not growing downwards towards gravity. It's moving away from gravity. And that's why we say that stems are negatively geotropic. Okay? So stems are positively phototropic because they move towards light as a stimulus. But stems are negatively geotropic because stems do not grow down towards gravity. They are not stems are not moving towards gravity. Therefore, they are negatively geotropic. Right. And here you can see how the stem is growing upwards towards light. So what is tropism? Tropism is a growth response, a bending movement of part of the plant towards a stimulus. So when the plant bends towards a stimulus, that bending is called tropism. Bending towards light, phototropism. Bending towards gravity, geotropism. Okay? So let's look again at some highlights on phototropism and geotropism. You can see that plant hormones cause stem elongation, and they also cause the germination of seeds, and they promote flowering. So which hormone exactly causes the stems to elongate? Now you find that you can have a stem and you have little swellings called nodes, okay? So this region between two nodes is called your internodes. Now the hormone gibberellins is actually going to cause the elongation, this area between two nodes. The cells will start elongating and the nodes will now move further and further away, and it's going to cause the stem to now elongate. Okay, so the hormone that actually causes your stem elongation, your seeds to start germinating like these seeds are germinating, right? It's going to cause the flowers to actually bloom, or sprouting of flower buds, germination of seeds, elongation of your stems. And the hormone that's going to do that is going to be called gibberellins, all right? And here you can see now, here you have your stomata, right, which is going to close in winter because you know that there's less water in winter. So the stomata is going to close, so you're going to prevent the wilting of leaves. And here, this function here is caused by abscisic acid. So what does abscisic acid do? Abscisic acid causes leaves to fall. Abscisic acid inhibits the growth of seeds in winter. Abscisic acid causes the closing of stomata in winter so that you don't lose a lot of water because there's less water in winter. Abscisic acid also causes seeds to become dormant. So on this side you can see all the functions of abscisic acid: leaf fall, closing of the stomata to prevent the plant from wilting, from drying out. It inhibits the growth of plant in winter. So plants don't grow. You'll notice that in winter you don't even mow the lawn much because the growth is very, very slow in winter. That's because of abscisic acid. Okay, so it inhibits the growth of plants in winter. Not all plants, but some plants. It also causes seeds to be dormant, Dormant means it won't germinate, and that's because of less water and it's very, very cold. So if the plants seeds had to germinate, the plants will die. So abscisic acid has a very high concentration in your seeds and plants during winter and causes the leaves to fall and your seeds to become dormant and your stomata to close. Right. Now, what does abscisic acid do again? It causes the seeds of plants to become dormant in winter because there's a lack of water, and it causes stomata to close so that the plant does not wilt. Okay. And here you're going to look at now again your phototropism and your geotropism. They can ask you in the exams to tabulate the differences between phototropism and geotropism. Right, and here you can see when the stem is exposed to light from one side, right, that is called unilateral light. Then the stem is going to bend towards light. Also, we're going to see now how do auxins actually behave. Right, now I'm going to draw a little stem here, right, and if I give the stem light from one side, auxins are very, very sensitive to light. Actually, light actually destroys auxins, so they're sensitive to light. So when light comes from one side, they actually move to the part where there is shade on the darker side. That's how auxins behave. They are light sensitive. When light comes from one side, they will move to the opposite side where it's shade or dark. And then on that side of the stem, that's the stem, the high concentration of auxins on the shaded side of the stem. See? A high concentration of auxins on the shaded side of the stem will cause rapid growth, rapid cell division, rapid cell elongation, and the stem on the shaded side will grow faster and faster and elongate faster compared to the side where there's light, and then the stem is going to bend towards light. That is called positive phototropism, or that's showing you phototropism. So when the stem is exposed to light coming from one side, the auxin concentration will be high on the dark side, like that side, because light destroys auxins. Where there's light there, more growth, you have to say more growth occurs on the dark side because the auxins will stimulate growth on the dark side. How does it stimulate growth? It will increase cell division and increase cell elongation on the shaded side, and the stem will bend towards light. Okay, now let's look at geotropism. So when the plant, actually the pot plant, right, it fell on its side. So what's going to happen is that when the plant falls on its side, remember now, here's your roots in your soil. So let us look at the roots. I'm going to look at the root, right? So here's your roots. When the plant falls on its side, now gravity is going to pull the auxins in the root down. So all the auxins are going to go on the lower side of the root. So here's your auxins. They are now on the lower side of the root because gravity is going to, that's not light. How are the auxins reacting now? They are moving or reacting towards gravity. So these arrows are showing me gravity. Here's your pot plant. It fell down. So here's your roots. So gravity is going to pull the auxins on the lower side of the root. Now learners, just understand that in a root, a high concentration of auxins is going to inhibit growth. Where there's a high concentration of auxins on the lower side, growth is going to be inhibited. And on the opposite side, where there's less auxins, so where there's a low concentration of auxins on the root, their cell division and cell elongation will be stimulated, and the side where there's low concentration of auxins in the root will grow faster and faster due to cell elongation and cell division occurring rapidly there, and the upper side of the root will grow faster than the lower side, causing the root to bend down and move towards gravity, and that is called now positive geotropism. And here you can see it. When the root is placed horizontally, you see like it fell down, then only one side is exposed to gravity, this side. The auxin concentration will be high on the lower side, you see, because gravity pulled it down on the lower side. Gravity attracts the auxins. Then there's going to be more growth on the upper side. So in a root, high concentration of auxins in a root inhibits cell division and cell elongation. Growth is slower, but where there's a lower concentration on the opposite side of the root, on the upper end of the root, then cell division and cell elongation will be faster, and the root will bend and grow down towards gravity. So the auxins on the lower side, right? More growth occurs on the upper side of the root because auxins on the lower side will actually inhibit growth. So on the lower side, growth is inhibited, but growth is stimulated on the upper side, and the root bends downwards towards gravity, and that is called positive geotropism. And here you can actually see again that this is just another picture showing you that here's light, and this stem is growing upward because when light comes from all sides, the auxins will move from the tip down in the middle of the stem, and then there's going to be rapid cell elongation and cell division in the middle, and that stem is going to grow straight up because now light is coming from all sides. So when your trees are in the middle of the ground with no walls, there's no buildings, that means light is coming from all sides, evenly distributed. What auxins do, they move from the tip downwards towards the middle, and they're going to cause rapid cell division and rapid cell elongation from the middle, and then the stem is going to grow straight up. Right, and here in this picture here, you can see light is coming from one side. So when light is coming from one side in the stem or the shoot, what did we say? Auxins will move onto the shaded side. Here you can see a lot of high concentration of auxins on the shaded side in a stem will stimulate cell division, stimulate cell elongation, and the shaded side will grow faster compared to the unshaded side where it is lit, and therefore the stem is going to bend towards light. And here is the root. Now this is the plant that fell on its side. It's horizontal, and what's going to happen here, when it's horizontal, you're going to find here the auxins will move on the lower side of the root. High concentration of auxins on the lower side will inhibit cell division, inhibit cell elongation, and promote faster cell division and cell elongation on the upper side, and then the root is going to bend and move down towards gravity. So this diagram here is showing you positive geotropism, and this diagram is showing you positive phototropism. Okay, and then you're going to see now here there's an experiment, and you have what we have, a clinostat. A clinostat is simply it looks like a clock with a little thing, that little structure that you can wind, and you're going to put this pot plant there, and you're going to put a little box over the plant. It's a well-watered plant. Now you're going to put a box. You're going to make a small opening in this box, and you're going to allow light to come through. But now in this experiment, right, in this experiment, this clinostat is stationary. You're not winding it. It's standing still, and light is coming from one side. So what's going to happen here? Because light's coming from one side, what did we say? Auxins are sensitive to light. They're going to move to the shaded side of the light, because if they stay near the lit side, they're going to be destroyed by light. So because of that, auxins are sensitive. They move to the shaded side of the stem, and then on the shaded side, they're going to stimulate cell division, stimulate cell elongation, and the shaded side will then grow faster than the lit side, and it's going to bend towards light. So this is called the experiment because it's getting light from one side, okay? And it's not wound out, so it's getting light, and you're going to see the bending movement, how the shoot or stem bends towards light. It's showing you the bending tropism towards light. And here is the control. So this control now, we are actually causing the plant to rotate. So here, this control has the clinostat that is actually rotating. Okay, so you're still getting light from one side, but now you're rotating. So all sides of the stem is going to receive light. So you're going to say this stem or shoot is going to get light from all sides. And what's going to happen? The auxins at the tip will actually move at the tip because light is coming from all sides now. Remember, you're rotating all sides. So auxins will move from this side, they'll move that side, because it's coming from all sides. They're going to move towards the center, and they're going to cause upward growth. So in the control, this plant is growing upwards. Can you see that? Straight up. Whereas here, because the clinostat is standing still or stationary, it's getting light from one side, the stem is bending towards light. Right, so that is the results of this plant. And you can see the experiment. The clinostat is unwound or stationary, but in the control, the clinostat is rotating. When your plant is actually rotating, like here in your control, all sides are going to get light. But in the experiment, where it is actually stationary, only one side gets light. In your experiment, what will be the result? The stem bends and grows towards light source. That is showing you positive phototropism. And in the control, because light is coming from all sides, auxins move in the center and causes cell division and cell elongation upwards. In the center, evenly, the stem is going to bend and grow straight up. Right. Now here is an experiment showing you geotropism. So they took little seedlings and they germinated them and they put them onto this clinostat. So this clinostat now, it is rotating. It is rotating, okay? It's just rotating like that. And they pinned little seedlings, germinating seedlings. So if you are rotating, you are eliminating gravity. Now, gravity can't pull you down because you are moving like this. So gravity is unable to pull the auxins only on the lower side. So what happens is that these radicals, remember the radical develops into a root. These radicals or developing roots will grow in all directions because there is no gravity. So they will grow in all directions. And this one here now, we pinned all these germinating seeds and it's unwound. Unwound means it is stationary. So it's not spinning. This one is spinning. This one is stationary. So here now, if you're going to put your germinating seeds, you're going to have gravity because it is standing still. So what's going to happen in this seedling, where this is a root, the auxins are going to move on the lower side. Then they're going to inhibit growth on the lower side, and the upper side in a root, there's going to be cell division and cell elongation, and the root is going to bend down towards gravity. See, even this one is bending towards gravity. That's bending towards gravity, so in the one seedling where it's not wound, there is gravity, and gravity is going to cause the auxins to move on the lower side of that root, and then the upper side is going to have cells to elongate faster, and the root is going to bend down towards gravity. Where? In the unwound, in the stationary clinostat. That's the experiment. And here you can see geotropism, positive geotropism here in that experiment. So that's a control. And in the control, you can see that it is rotating, you're eliminating gravity, and the little radicals or roots grow in all directions. In the experiment, it is stationary, it's not wound, you have gravity, gravity will pull the auxins down, and in a root, the upper side will grow faster than the lower side in the root, and then the cells will grow faster on the upper side, causing the root to bend and grow towards gravity. Right, and then we're going to look at your thorns. Your thorns are actually a defense mechanism, so you can see your roses. Why must they have thorns? Because they don't want anyone to take the roses off before there's pollination. So it's a form of defense mechanism. Otherwise, if you break the roses, before they can be pollinated, roses can become extinct. So the thorns are very sharp just when the rose buds are going to be blooming, because what happens? The plants are going to defend themselves against predators, against humans as well. So this is called a defense mechanism. It's a thorn. It's a sharp structure for protection against herbivores so that animals do not eat them, and also to prevent the flowers from being broken before they can actually get pollinated. So you can also see on the cactus, right, that this cactus has sharp spiky structures called thorns. It's also to protect the plant so that the plant is not damaged before they can actually flower and fruits can form. Here you can see the acacia trees as well, that they are protecting the plant so that the herbivores don't eat up the leaves, because if they eat up the leaves, then there won't be any structures to photosynthesize and the plant can die. So therefore, these acacia trees have small little leaves, and the herbivores love to eat small leaves, but if their thorns are there, they're going to protect it against the herbivores. So basically, learners, thorns are protective structures in a plant because the plant cannot fight. So the only way it can defend itself is by using thorns. We also have some plants that give off a pheromone, or like a smell or a chemical, and that chemical will actually repel insects and repel certain herbivores from eating them. So plants have adapted to prevent herbivores from eating them because they will release or secrete a chemical that can be very bitter, so the animals don't eat them. So these plants produce chemicals called phytoecdysteroids. You don't need to know that full name. Phyto always means plant. Ecdysteroids are the chemicals of the plant. So plants produce chemicals, right, to prevent the herbies from eating them. So these chemicals are very, very bitter. They also can have a very strong smell to actually repel the insects and repel the other animals. So the chemicals cause the insects, if the insects should eat them, these insects now will molt and they will die prematurely. They will molt very, very, very fast and eventually die. So therefore, these chemicals act as repellents for the herbivores so that they won't be eaten. So you can see tobacco plants also give off a very pungent smell so that nothing can eat the tobacco plants. Remember, your tobacco or your cigarettes are made from leaves. So if animals or herbivores have to eat them, then there wouldn't be any tobacco. Therefore, the tobacco plants gives off a pungent smell, and that is a chemical for defense. Right. Also, the mopani tree, you can get mopani worms that can feed on them. So these mopani trees give off a very aromatic or pungent smell to repel certain insects from eating them. And we can just look at some terminology relating to what we've just done. So give the correct biological term for each of the following. Write down only the term next to the number. Cell that stimulates cell elongation on the shaded side, right? So what will that be? Actually, it's supposed to be hormones that stimulate cell elongation on the shaded side. It's going to be auxins. Plant substances that act as a growth inhibitor. It's abscisic acid. Remember, abscisic acid causes leaves to fall, seeds to be dormant, and plants cannot grow in winter. So that's abscisic acid. And what do we call the movement of a part of a plant to its stimulus? General movement or bending. It's called a tropism. And the tissue in which auxins in plants is mainly produced, it is called the apical meristem. Now remember, apical meristem means the tip of the root and the tip of the stem. So the growing tip here, that is the apical meristem, and the growing tip of the root, that is the apical meristem. That's where the cells are actively dividing on that tip, the meristematic tissue, and that's where the auxins are found. Where? On the apical meristem of your stem and apical meristem of your root. Right. So give the biological term for the movement of roots downwards. If roots move downwards, they're moving towards gravity, and that's called positive geotropism. A growth response of a plant towards light, Phototropism. The main hormone controlling leaf abscission means leaf fall, abscisic acid. And the apparatus which is used for demonstrating geotropic growth to show that roots grow down towards gravity is going to be a clinostat. That is a rotating structure, okay? And learners, please understand that we are going to now go on to questions, and my colleague, Mrs. Segeka, is going to come through and do all the questions relating to plant hormones, but also know that it is the gibberellins that causes seed germination, sprouting of buds, and elongation of the internodes in a plant. With that, thank you, learners. Please learn your notes and apply all this content to the investigative questions.
Right, welcome back, grade 12s. My name is Sina Sekheche, and I will be taking you through the questions that you may come across on this wonderful topic. And thank you so much to my colleague Suki for taking you through all these wonderful pointers that you need to know on this topic. So let us get into the questions. Remember, this topic mostly you will find, or in this topic mostly you will find questions or investigative questions. So you must be ready to read with understanding. Take your time, underline, understand what was done in the investigation, read your results and interpret the results, and then be able to answer the questions. So don't just rush to answering without understanding what is happening. So we're going to do that with the questions that we have. So these questions are taken from various question papers. You can also do that when you are studying. Right, the first question we have here is from your prelim paper 1 in 2022. Let's see. A pot plant was placed onto its side in a dark box. Now this dark box is highlighted because then you would want to ask yourself, why was a dark box used instead of just on light? Because you want to eliminate light. You don't want light to be a factor in this investigation. Right. After 2 weeks, the stem had started to grow upwards. So we see here is a potted plant that was placed on its side. So you see here it's bending, it's on the side, it's laying down. Then after 2 weeks, the investigators observed that the stem, remember this is the part of the stem, this part of the plant is called the stem, is now starting to grow upwards. Now you ask yourself, what could have caused that? Having studied how auxins or hormones in plants act, you must think of what is happening in this plant. Then come up with an example. Suki just told you how auxins move to the lower part of the stem when gravity is at play. So in this case, you know that gravity is at play because this plant is put on its side. So when it's on its side, there is a part that will be at the bottom. So if there's a part that is at the bottom and top, the auxins, the gravity that is at the bottom is going to pull the auxins to the bottom side. So meaning more concentrations of auxins are going to be found where? At the bottom. And when they are found there at the bottom, you know that gravity auxins, when they're at the bottom, they are going to be inhibited. They are going to inhibit growth or cell elongation, meaning our plant on that side is not going to grow. But on the side where there is no gravity, what is going to happen? There is going to be more cell elongation, more cell division. Then with that cell division and cell elongation, what is going to happen? It is going to cause our stem to grow upwards. So this information is your background information that you've learned before or while studying. You did not go to the questions first to check. Now let's go to the question and see. Name the experimental factor responsible for the stem's upward growth when a pot plant was placed on its side. So you think, what made this stem to grow upwards, or what is that environmental factor that caused this thing to happen? It is gravity. And before you even ask this question, you knew because you tried to analyze this investigation on your own before you even go to the questions. Right, and then let's go to the second question. It says, no, identify the phenomenon indicated by the stem's upward growth. You cheated there, you got the answer before, but you still need to read the question so that you can understand why that was the answer. So the phenomenon that is indicated by the stem's upward growth, that is known as Geotropism, because gravity is at play here. Right, remember we are not talking anything about the light. Whenever you are reading an experiment, start there and check which factors are at play here. And you know, because light is not at play, there's no way you will talk about phototropism. However, you are talking about the stem's upward growth, then you know we are talking about geotropism. Right, name the plant hormone responsible for the stem's upward growth. It is the auxins. Remember, you just learned about the auxins activity in geotropism and also in phototropism, and they act differently. But in this case, we still know that the hormones that are responsible for stem's upward growth are the auxins. Right, these are your free marks that you get for your revision. This is the same experiment. Now we continue with the questions. We say, describe how the distribution of the hormone mentioned in question 3.2.3, meaning on the previous question you did mention the hormone which was the auxins. So now you must describe how it has caused the stems to bend in an upward direction. With the background information that you started with, you are not supposed to scratch your head because already you know that more hormones, but in this case you know that it was the auxins. But if you wrote hormones, let's say you got it incorrectly in the previous question, you wrote gibberellins or abscisic acid, but here you would say hormone. The examiner might say they will give you a mark for not knowing, they will not penalize you twice for not being able to mention that hormone in the previous question. Hence we have more of that particular hormone. So, but in this case, we know because we've already mentioned it, it is the auxins. So what is going to happen to those? They are going to accumulate, or they're going to have a higher concentration on the lower part of the stem. You get a mark for just mentioning that. So you say here on the lower part, there's going to be more concentration of auxins. You get a mark for saying that. And then when there is more concentration of auxins, remember your notes. It is going to cause more growth or elongation on that side. Then fewer hormones or auxins are going to be accumulated on the upper side of the stem. Now you are telling us the opposite. Remember, when more are pulled by gravity to the lower part, what is going to happen to the upper part? The upper part is going to have less, meaning there's going to be growth where? The less growth on the upper side. And when there is less growth on the upper side, the stem is going to bend what? Upwards. Right. Explain one way in which the stem's upward growth benefits the plant. So if the plant is now growing upwards, remember the plant's needs. What does a plant need? And if it is placed on its side, it is going to strive to look for something that it needs. So what are those things that the plant needs? So how is it going to be beneficial for that stem to bend or grow upwards? Right, the first benefit would be the leaves of the stems are going to receive more sunlight, or they're going to face the light more for what? For photosynthesis. You know, plants, they manufacture their own food using sunlight through this process. So sunlight is quite beneficial for the plants. Hence, if the plant maybe on the mountains, you see the plants on the mountains that are growing, remember the mountain is uneven. There are other sides where the plant will be growing from the side of the mountain, then the stem will be bending upwards. That stem is bending upwards to make sure that the plants receive sufficient sunlight for photosynthesis. Or you can say that it is going to face the light for photosynthesis, or you can say that it exposes the flowers in a more favorable position for pollination or for seed dispersal. Remember, you want your plants to keep on pollinating to form other species, right? So we are going to take a break from this question, and then we will continue with the next question after this. Right, now we continue with the question. We continue to the next question. Now this question was taken from the June paper 1 for 2025. So this is quite a recent question paper. And remember what I said to you, you have to be prepared to read with comprehension. Now let's take a look at what is happening here, because most of the questions in this topic are an investigation type of questions. Right. An investigation was conducted to determine the response of plant seedlings to light. So already you now have, you see, they have to respond to light. So what happened? How did they conduct this experiment? The procedure was as follows. Grain seeds were allowed to germinate into seedlings. So now you have seedlings that have been germinated. 1/2 of the seedlings were planted in one small pot. So you have one small pot, then another half in another small pot. Right. So you have half, right, of what? How many? Those seedlings. Now, when the stems reached a height of approximately 1 centimeter, so these are the seedlings. If you don't know where the seedlings are, look at these structures here. These ones, I don't want to mess them a lot. They are your seedlings. Those ones that look like your matchsticks, they are your seedlings. So when they were about 1 centimeter, what did they do? The pods were placed on the discs of 2 clinostats. You've just learned what is a clinostat. So here in our case, this is our clinostat. So 1/2 was placed on a clinostat and another one on a clinostat, right, on the discs. So these are the discs. These are the discs of the clinostats. Right. A clinostat is an apparatus consisting of a slowly rotating disc when switched on. So that is just a background to explain to you what is a clinostat. Now, both sets of apparatus A and B were placed inside boxes with openings on one side only for light to enter, meaning this is light coming from one side. We call it unilateral light because it's coming from one side. Uni means one. Right, so let us take a look at where the light is coming. Here is the opening, light. So you follow. Seedlings placed on the discs. After being placed on the discs, and 2 boxes were opened on the side. Each box covered each setup. Right. Then one clinostat was switched on. One was switched on and the other one was switched off, meaning when the clinostat is switched on, it is rotating. That disc will be rotating. The other one is not rotating, but at this point we don't know which one was switched on, which one was switched off between the setup of apparatus A and the setup of apparatus B. The apparatus was left for 2 weeks. So in these 2 weeks, what do you do as a scientist? You are observing. So let us check what is it that we are observing. In apparatus A, you can see that the seedlings are starting to grow and bend towards that small opening where light was coming. However, in apparatus B, the seedlings are growing evenly upwards. So this is the information you have so far from your observation. Then let us go to the questions. Identify the plant growth responsible or the plant growth response demonstrated by the results. So what is the plant growth response there? It is phototropism. Why phototropism? Because we want to see their growth in response to light. Right. In which apparatus, A or B, was the clinostat switched on and the discs rotating slowly? Let's go back. Let's see. So between A and B, which one would you say the clinostat was rotating and which one was not rotating? Because you see that this one, the seedlings are growing evenly upwards. You assume that they were receiving light from different sides because it was rotating. Because they are not bending towards any source of light. So they were able to get light evenly. That even part is brought by what? By a wounded clinostat. Let's see if we are correct. Right. Go. B, which is correct. In our apparatus B, it is where our seedlings are growing straight upwards. So we assume that because they were growing straight upwards, the clinostat was rotating, it was switched on to allow the seedlings to receive light evenly. Right. Explain your answer. Let's see. All the seedlings grew straight. We saw that. You get a mark for saying that. Do not be lazy to write what you think, because sometimes you may find your answer is correct, but you are thinking that it might be asked in a different way, or they might expect you to answer in a different way. So answer. So it showed no phototropism, or you saw there was nothing growing towards light. Remember we said that? Because they were evenly exposed to light, not exposed to unilateral light. So you assume that, okay, because they did not grow towards a particular light, it means they were receiving light evenly. We have already said that before even being asked the question. Explain the growth response of the seedling in apparatus A. Remember, in apparatus A, it is where the seedlings were growing and bending towards that hole where light was coming in. So tell us what was happening in the plant for that growth to be happening. Right. We saw that the seedlings or the stem, they grew or they bent to the left. Why were they bending towards the left? You see, you get a mark for that. It was just your observation. And then you say, because they were exposed to light from one side. Remember that light was coming from that opening on the box. So it was unilateral light. There was no light that is coming from any other source. Then the auxins, now you are telling us what was the effect of that. The auxins diffuse to the right. Remember, auxins are sensitive to light. So if the light is coming from the left, what is going to happen? The auxins are going to move to the dark side. So they are going to move to the dark side where there is no light. So the dark side of the seedling will be on the right, whereas the light was coming from the left. Right. Now, the higher concentration of the auxins on the right or on the dark side stimulates cell elongation, yes, on the right of the plant. Remember, it is dark. Now the auxins have moved towards the dark. When they are there, what are they going to do? They are going to stimulate cell elongation on that side. And when that cell elongation is being stimulated on that side, it is going to make the stems to grow more, and as they grow more from one side, it causes them to bend towards light. So that is the effect of what is happening in that experiment. Then we continue to look at another one where we are learning about the roles of auxins again. So the learners here, let us check what they did. A group of learners conducted an investigation to determine the effect of auxins on the growth of stem in bean seedlings. So here, they did not talk about light, they did not talk about the dark box. So it's just the effect of auxins on the growth of stems in bean seedlings. So let us check what they did. The procedure was as follows. 30 bean seeds. Remember, in an investigation, if you are given a number, it is not a number for you to just go through it and then not put much concentration on it. You need to think of it is the number that is you're going to talk about this number, increasing the sample size for your reliability. Remember, but you must be specific and talk about the number, because when you increase the number or increase the sample size, you must tell us what was the initial number and how much are you adding or how much do you want them to add. So consider this number when you are answering that type of a question. So going back to the procedure, 30 bean seeds were allowed to germinate for 5 days to produce seedlings. Also, this time, also very important for you to talk about when asked about that question. Now, the seedlings were divided into 3 groups. Remember, we had 30. When you divide them into 3 groups, it means you are going to have 10 in each group. So group A had 10, B and C of 10 seedlings each. The tips of all the seedlings were cut at the same length. Follow all the 3 in each group, the tips were cut at the same length. And then in group A, the cut tip was placed back on top of the young stem, meaning they cut it in group A and they put it back. Then in group B, the tip was not placed back, meaning group B, it was left without a tip. It was just a cut open stem. In group C, the piece of plastic was placed on top of the cut surface. Follow. They took a plastic and put it on top of the cut surface. And then the tip was then placed back on top of the plastic. So it is the stem, it was cut, they put a plastic and they put the tip again on top of the plastic. So that was in group C. So the growth of the stems was then observed. When we look at the diagrams, they're going to explain to us very much easily. You check group A, we said that the tip was cut then replaced. So you just cut it and put it back. But in group B, it was cut and not put back. So it is open. But in group C, it was cut, a plastic was placed there in the middle, and then the tip was put back. Now, you are thinking of the roles of the auxins. Remember, you find the auxins there at the tips. That is your apical part. So you find your auxins. So you think, if you remove that part, what is going to happen to the auxins? So let us look at the questions. Identify the dependent variable in this investigation. So what is dependent here? Stem growth. So the stem will grow, but the growth of the stem is dependent on something which we've removed or which we have placed. So stem growth, not only just the stem growth is dependent on what? On the amount of auxins or on whether the auxins are available or not available. So you see, that's how you can determine your dependent and your independent. Now, why did the learners cut the tips of the young stems? So why were these tips cut? Because what do you want to remove there if you are cutting this? Remember the aim of the investigation. We were checking the roles of what? Of auxins on stem growth of these seedlings. So they cut those tips because they wanted to remove the source of auxins. Remember, the auxins are found there at the tip. So you are removing the source of the auxins. So because that is the element, that is the factor you are investigating. Then the tips are the ones that produces auxins. So that is any one of those you can give and get a mark. Now give one reason why 10 bean seedlings were used in each group. Why not 1, 2, or 3? Why did they use 10 in each group? To increase the reliability. Remember, the more you do, the more your results are becoming valid. So the results are valid, but it increases the reliability of those results. Then write down the letters of A, B, or C of the groups where there will be no upward growth of the stem. So let us take a look at A. A, this is the source of auxins. The tip was cut, but the source of auxins was put back directly on top of the stem. In B, the source was removed completely. In C, the source was removed, a plastic was placed, and the source was put back. Now here you see there is something that is stopping the source to get here, to get to the stem. Here there is no source at all, so it means there is nothing that is going to move to the stem. Here the source is there and there is nothing that is inhibiting the flow of the auxins from the tip to the middle of the stem. So in which one of these there will be no upward growth? In B, we said there is no upward growth, and also in C. Why? Because in B there is no source. In C, the source is blocked. So there is no way that the stems are going to grow upwards, because in order for them to grow upwards, what do you need? You need the source of auxins, which is the tip in this case, to do what? To release the auxins evenly downwards, because these seedlings are receiving light from So all around evenly. You want them to, you are expecting the auxins to move where? To move downwards, and as they move downwards, they are going to cause what? Elongation, and the stem will grow upwards. So that is going to only happen where? In diagram A. However, in diagram B, there is no source, there are no auxins that will be moving downwards. In diagram C, the source is blocked with a plastic. So there is no transfer from the tip to the stem. I hope you understand how we got to that. Excuse me. Continuing with the same question, describe how auxins cause apical dominance. Now, apical dominance, you spoke about this with Suki earlier. Let us see. The presence of auxins in the tip of the stem is going to stimulate the upward growth, and it is going to inhibit the development of lateral branches. So when it is apical dominance, it is allowing the plant to grow upwards. And if the auxins are there, they are going to inhibit our plants to grow lateral branches, meaning on the sides, to grow on the sides. So this shows that the auxins are dominant for growth that is going upwards under even light. But then if they are there, they are going to inhibit. To inhibit is to stop. They are going to stop or to reduce the growth of lateral branches, or the branches that are on the side. So that is the apical dominance of auxins. Right. Name the plant hormone other than auxins that promotes the germination of seeds. So what also promotes? Remember when you were doing the functions of these hormones that you've studied today, you saw that other hormones share the functions. So what other hormone is responsible for the germination of seeds if they are underground? It is gibberellins. And remember the spelling, as Suki told you, that the way you sound it, it has got two B's, like gibberellins. So please make sure that you remember the spelling when you are writing this hormone. The hormone that inhibits the germination of seeds. Now this one is to promote, we want more. To inhibit, to stop. So these words, it is acting now in opposite. So which hormone is now going to inhibit the germination of seeds? Remember, Suki was also telling you about winter. What is happening to your grass in winter? It is not growing more. So what is happening? Or if you plant something in winter, it will not grow because it is already the mechanism of the plant to say you can't grow in these cold conditions, because then the plant is going to die or it's going to fail. So abscisic acid will be found in more concentration in those types of climates. To do what? To inhibit the germination, because the environment is not favorable for that plant to grow. So that is quite amazing how it happens. Then we can look at another experiment where we are still investigating further the role of auxins. So let us take a look at what we are doing in this investigation. Remember, these were taken from previous question papers. You need to read with comprehension. An investigation was done to determine the effect of a plant hormone, not mentioned, see, on plant growth. So the procedure was as follows. 2 pot plants, 1 and 2, of the same species, very important also, because you need this to answer some of the questions in your investigation, and age were used. So you have the same species and age were used. The apical buds of both plants were cut at the same length along the stem. So when you look here, you see this is your stem in your pot plant 1. The apical bud was cut. And also in pot plant B, the apical bud was cut. Right. The cut surface of plant 1 was sealed with petroleum jelly. You need to stop and go back and understand. So you have the pot plant 1, they said they cut the apical bud, or the tip. You can say it's also the tip, but here they are calling it the apical bud. Now, after cutting the apical bud, we are told that that opening of the stem was sealed with petroleum jelly or Vaseline. Then The cut apical bud of pot plant 2 was placed on a block of agar jelly for 2 hours. Follow. Remember, if it is an apical bud, they cut. If it is a stem, they cut the apical bud. They take that apical bud of pot plant number 2. They put it in a block of agar jelly. They leave it there for 2 hours. Right. Then the block of agar jelly was then placed on the cut surface of pot plant 2. Then after 2 hours, they go to that pot of agar jelly. They take that block of agar jelly. They put it back on top of the stem of pot plant number 2. I hope you are following. You have this in your mind, you can visualize it. Right. Then the plants were exposed to the same environmental conditions for 2 weeks. Meaning light, water, whatever that the plants need, they were exposed to the same environmental factors. Now the growth of both plants were observed at the end of this period. The diagrams on the side are showing this. So you see in pot plant 1, there is growth, but it is not as high as in pot plant 2. Fine. And also you see in pot plant 1, there's something that is different. You see there are lateral branches, but in pot plant 2, there are no lateral branches. Already you are thinking in your mind, what is the role of auxins? You've learned that. What is the role of what is the role of auxins? Let us take a look at the questions. Now the first question says, state why the apical bud was placed on a block of agar jelly for 2 hours. Remember, they cut the tip of pot plant 2, of the stem of pot plant 2. They put it in agar jelly, that tip. They put it on a block of agar jelly and left it for 2 hours. Why were they doing that? What is on the tip? It is the auxins. So they did that so that the plant hormones that are found there on that apical bud, or the auxins from that apical tip, they could diffuse into the agar block of jelly. So they wanted that those auxins to diffuse into the block of jelly, meaning the block of jelly is now going to do what? To have the auxins. Right, so they wanted to do that. Describe the results obtained for pot plant 1. So pot plant 1, what are the results here? What do you see? The stem stopped growing upwards. You see this stem here? It stopped. It is shorter. Why do we say it is stopped? Because it is shorter than the one that is on the right. So you assume that the stem stopped growing. Then the lateral branches started to develop. That is just an observation of the results. You are not interpreting. But at the back of your mind, you must have an explanation to say, why? Why did the lateral branch form? Remember the apical dominancy of auxins. But here, because we cut the tip and we just blocked it with petroleum jelly, there are no auxins coming in. Because the auxins inhibit lateral branches from forming. In this case, because they are not there, this is going to allow the lateral branches to grow, to form. That is your background information on the activity of auxins when they are at the tip. You think about the apical dominance that we spoke about. Now, explain how fruit farmers can use the knowledge from the results in question 2.4.2 to their benefit. So, if farmers know that if they remove the tips and they remove the auxins, their plants are going to grow lateral branches, how is that going to help the farmers? Right. The lateral branches are going to develop, and that can bear more fruits. Remember, if we have more lateral branches, each branch as it branches out, it creates more surface area for fruits to grow. So it means this can leave farmers with more fruits that can also increase what? The yield when they go and collect. Or you can say that the shorter trees, remember these trees, because the auxins are cut there at the tip with the source, they are going to grow now to the side. It means they are no longer going to be elongated upwards. They are going to grow more on the side. And as they grow on the side, it means they are shorter. So we are going to have shorter trees or the development of lateral branches, which is going to make harvesting of fruit easier. So the people who are going to be picking up those fruits, they are going to be within reach because the trees are shorter. So this information can be very beneficial to the farmers if they use it and apply it, meaning with all their crops, if they cut the tips, the apical tips, they are going to increase what? The lateral branches for growing, and that is going to be beneficial to them to make sure that their trees are short, and during the time of harvesting, the fruits are within reach. Or we can have more fruits with more branches. Then it can increase what? Yield. I hope it makes sense to you and to the farmers, right? Explain why the stem in pot plant 2 grew upwards. Now you see in pot plant 2 here, it grew upwards. This takes you to remember what happened here. Remember the agar block that was put on the side where the auxins diffused into it. That agar block was then placed on top of that stem. So what was happening there? The auxins in the block of agar jelly, they moved downwards. Remember this agar jelly, it has auxins. Where did it take these auxins from? From that tip that was cut. They allowed them to be on top of this agar jelly so that it can diffuse into the agar jelly. So this agar jelly has auxins in them. So when you place the agar jelly on top of the stem, as it melts, what is going to happen? The auxins are going to move evenly downwards the stem, and as they move into the stem, it is going to cause cell elongation there in the middle of the stem, resulting in what? In an upward growth of the stem. So that is the reason why this one kept on growing upwards, because the source of auxins was just changed from the tip and dissolved or diffused into the agar jelly or into the block of agar jelly. I hope it makes sense to you. Then another investigation that we can look on is the role of auxins in phototropism. We've already done this, but still, remember, you need to do more and more because you don't know which one an examiner is going to ask you. So you need to expose yourself to more questions and interpretation. This will allow you to interpret different types of questions, right? An investigation was conducted to determine the effect of auxins on direction of growth of stem. So we want to see if the auxins are there, and then where are the stems going to grow, in which direction are they going to grow, right? The procedure was as follows. 2 potted plants, 1 and 2, of the same species and age were used. The apical buds of both plants were removed by cutting at the same length along the stem. So this one is similar to the one that we just did. So when you look at the diagrams, you need to be able to interpret the diagram. Sometimes they can show you light from the right or from the left. You need to be able to interpret and apply your knowledge. So you see here, it was cut on pot plant 1, and on pot plant 2 also it was cut, right? The cut apical buds of both plants were placed on a separate blocks of agar jelly for 2 hours, meaning those 2 cut stems were separated, or they were put on separate agar jellies, right? 1 block of agar jelly was then placed on the right hand top edge of the cut surface of the pot plant 1, right? Do you see here on pot plant 1, follow, the agar jelly is placed where? On the right hand top edge. It is not placed right on top of the middle of the stem. It is placed towards the right. Are we following? Right. And the other block jelly was then placed on the left hand top edge of pot plant 2 as shown in the diagram alongside. So here, this one, meaning 2 agar jellies, they are richly supplied with auxins because the tips of these stems were put on those agar jellies. Now, the placing of these agar jellies, they are not placed directly on top of the stem. The one is placed in pot plant 1 is placed slightly to the right, and the one in pot plant B, the agar jelly block, is placed slightly to the left. We are following. Both pot plants were kept in a dark box for 2 weeks. There is no light that is coming in here. You are eliminating. Remember, once you talk about a dark box, you are eliminating light as a factor that may contribute to the growth that you are investigating. So here there is no light that is coming in. The direction of growth of the stem of both plants was observed at the end of this period. So the direction of the growth was observed. Now, give a reason why the apical buds were removed. Those apical buds, the top parts, the tips, why were they removed? What do they have? To remove the auxins that are produced at the apical buds. So that one you must remember. Why were the apical buds placed on the blocks of agar jelly for 2 hours? Those apical buds that were cut, they were placed on agar jelly. Why for 2 hours? Because you wanted the auxins to diffuse into that block of agar jelly. You want the agar jelly to possess or to contain, to have the auxins. That is why you put the tips on top of the agar jelly. So the agar jelly will therefore have the auxins that are found in the tips. That one, please don't forget it. Now, pot plant 2 was exposed to unilateral light from the left side by removing the lid on the box during this period. Right, you see here on pot plant 2, here where I'm circling, there is a lid towards the right or the left side. It is coming from one side. So this was removed. When it is removed, it allowed light to come into that box. Now we have introduced a factor where light is going to, or a light, or let's say a factor which is light now. So this plant, we want to see how is it going to behave. Explain what would be the results if that happens. Right. If light is coming from that side, that one side, the lid is open, coming from the left, the auxins will diffuse to the lighter side or to the left hand or move to the darker side, meaning they are going to move away from light because they are sensitive. Don't use this diffuse because sometimes it might confuse you about the auxins. Just say they are going to move to the darker side, which is something that is basic that you know, that when light comes into a plant, auxins are sensitive to that light and they're going to move away from that light, meaning they're going to move to the darker side. Just state it like that. It is simple when you say it like that. Then, high concentrations of auxins at the darker side is going to stimulate cell elongation. So that darker side where the light was not coming, cell elongation is going to be stimulated, or there's going to be more growth of cells on the lighter side. Then this uneven growth, you need to say that it's uneven because then we need to see the bending. It is going to cause the stem to bend to the left side or towards the light because the growth was way more on the darker side. So any three of these where you will get, and then there's one which is your compulsory mark where you were supposed to say the bending is going to be towards the lighter side, not on the darker side, because the darker side is where your auxins will be more concentrating and causing the bending towards the lighter side. So that was a compulsory mark that you needed to say in whatever that you are going to say in your answer. We needed to see that this stem is going to grow towards that light. Right. Right. The last question here. Explain why fruit farmers periodically remove the apical buds from the fruit trees. We've already learned about this. So every now and then, the farmers go and they remove or they cut the apical parts from the fruit trees. What do they not want? What do they want to see? We've learned that. So you see, it is asked in a different way, but the background information you can also apply is the same. More lateral branch is going to develop. Remember, we are eliminating the apical dominancy of the auxins. We don't want our shoot to grow taller and taller, but we want to develop what? More lateral branches, which is going to increase yield. So for the farmers, this is good news. If we have more lateral branches, we are going to have more yield in terms of fruit, because they are growing fruits. So we're going to have more fruits, which is more yields. The fruit will be accessible from the branches, enabling easier harvest. Remember now, the trees are now shorter. They are not growing up and up and up and up uncontrollably because the auxins are cut. The lateral branches are now making it easy to reach for the people who will be harvesting. So that is the reason there. You see, it was in a different way. Please continue with your worksheet. Continue to answer questions on this topic. Here is a question where you are investigating the role of auxins in geotropism. This is the key thing that you need to know. When a plant is placed on its side, how is the growth going to be? The growth of the stem is going to be upwards. Where is the concentration of auxins going to be more? More concentration of the auxins is going to be on the lower side, being pulled by gravity. Then how is the growth going to be? The growth is going to be more where? On the upper side, because it is inhibited. And when it is more on the upper side, it's going to cause the stem to bend upwards. So if you know that for geotropism, you will be able to answer any questions on geotropism. Then these are the questions that I would like you to go through on your worksheet. Please make sure that you cover the entire worksheet. This is another one that you can go through. Here you check also our stem is bending downwards. You had this one in your notes, something that is similar to this, that is showing you the stem that is bending downwards. Know the reasons, know the position of your auxins, know how or what is going to be the effect of them being in that position and what puts them in that position. For geotropism, you would say they are going to be, gravity is going to pull them towards the lower side. For light, you're going to say they are sensitive to light, they will move to the darker side, and know how they are going to act whatever position you find them. So you go through this question also through your worksheet. Please go through your worksheet and answer this question. Now, there was a last part where you were learning about the control of weeds. Here, let us look at this problem just to give you understanding, and then you can answer the questions. Weeds are problematic to farmers, so no farmers want weeds because they invade farm yields and outcompete crops for space. So they take out the space for that. This reduces crop yields. Farmers spray their fields with chemicals known as herbicides. So you kill these weeds for herbicides because they outcompete the yield that you want to grow. To kill these weeds. Now, some weeds, however, have evolved to be resistant to herbicides. When we say they are resistant to herbicide, we mean that even if you spray the herbicide, it will not kill those particular types of weeds, meaning they will still survive. Scientists investigated the time it took for species of weeds to develop resistance to 5 types of herbicides. Right. So here are the 5 types of herbicides. Here is the time taken for those weeds to develop resistance, meaning after 9 years, when you spray the weeds with a 2.4D herbicides, it will not kill it. After 9 years, if you spray the weeds with Dalopon, it will not die. This is what the table means. So you need to be able to interpret that table. Now, refer to the passage. Okay, you read these questions You answer based on the time it took for these weeds to develop. What is more important is you need to be able to have the skill to draw a graph. When you have data like this, be able to analyze your independent variable, your dependent variable. So if you are asked to draw a graph on those results, to say, draw a bar graph to show the time taken for the evolution of resistance. Now, it took 9 years for 2.4 for the weeds to be resistant to the 2.4D. So what are you going to put on your independent variable? Independent variable comes on the bottom or on the x-axis, which is your herbicide, and then on your y-axis it is the time taken, how long it is, dependent on the herbicide that you are giving. So you are going to put that on your independent or on your dependent variable, which is your y-axis. Then here you have your different types of herbicides and the time it took weeds or each weed to grow resistant to these. Remember, when you draw a graph, draw this, write the criteria for yourself to check if you have put everything in the graph that is required for you to get the mark. And the heading of the graph is also important because it needs to have both these variables. So time taken for the evolution of resistance to different types of herbicides. So you see you have time and you have herbicides here. Then when you read the graph, you will get the full information. Please go through these questions as you prepare for your preliminary examinations. And we are going to see you again next week on the 7th of August at 2:30 as we will be dealing with evolution theories. We will see you again next time, and thank you so much. Right, let's go through that.